This is the wonderful Bree. She's the model. So what we're going to do today is we're going to talk about the Staphylococcus and the micrococcus. These organisms, if you look at them, they're gram-positive cocci. If you look at them microscopically, they're in clusters or tetrads, but that's presumptive. What does that mean?
Assuming.
You assume. That means it's not perfectly true, but at least you have an idea. The next test is the catalase test, and that's definitive. What does that mean?
For sure.
It's for sure. That is the diagnostic trait of these organisms. So the Staphylococcus and the micrococcus are always going to be positive for catalase. Okay? And then you can see that all of these organisms that belong in these particular genera, the Staphylococcus and the micrococcus have got a name change. They're now Koshura. All of those organisms are all halophiles. What does that mean?
Like the heat.
Heat. Not heat. They like high salt concentrations.
Oh yeah, salt.
Okay? So, and then the major pathogen in this group is Staph aureus. That doesn't mean that the other guys aren't going to cause you troubles, but they're likely not to because they're with you all your life. If they get into the urinary tract or in the blood or in the skin, they can cause infections. The simplest infection that they can cause is a pimple. How many people knew that a pimple is an infection? It's an infection of a hair follicle. It is true. So you got a pimple, it's not because you ate a lot of chocolate or ate greasy food or anything like that. It just means that you got an infection of one of your hair follicles. Good? I know you're like, well, we're women and we don't have a lot of hair follicles on our face. You do. They're just not active, which is a good thing. Okay? So Bree and I are going to start with the catalase test. And so Bree is going to put a drop of hydrogen peroxide on each one of these slides.
Can I leave it on the table like this?
Yeah. And you should. That's a perfect drop. Okay. Lisa, will you do me a favor? Will you bring me a plate of PEA, mannitol salt, and blood? Because it seems like it's gone. Somebody took them.
I took them.
I know you did. They're right there. You don't have to wear gloves to pick them up.
Do you want more?
No. She's gonna bring them. I just want to have them ready. I thought it was all set up, but it appears it wasn't. Thank you very much. Okay. So here, Bree has actually taken one drop of hydrogen peroxide on a slide that's labeled Staph aureus, catalase positive, and then the other one is Streptococcus pyogenes, catalase negative. Okay? So what she's going to do now is these are sterile loops, and if you pull these out of here, remember if you touch it in the middle but you don't touch either side, then the sides, the ends are sterile. So grab it from right here. Okay? And then I want you to go in and with the pointy side, go in here and just scratch some of the organism off of the surface, all the way down to, no digging. Okay, almost keep down a little more.
I'm scared to touch the other end.
No, you're good. No, you're good. Okay, pull it out. Okay, so this is Streptococcus pyogenes. So I want you to come over and be the witness. Okay, the other one of you. It's okay. So I want you to put that and mix it around in the one that says Strep pyogenes. And Marisa, you tell people what happens. What happens? Nothing happens. Okay, good.
I see.
Turn it around. Turn it around. And then I want you to take Staph aureus and I want you to do the same technique. Go in there and grab some stuff. Down here where there's, okay, good. I think that's it. So now, Marisa, come over. And Marisa's going to be the witness. Does anybody want to take a picture? Because it's going to be pretty. I mean, it's not going to take it's going to go fast. So, okay, so don't block the camera. Okay, so now watch what's going to happen. She's going to put it into there and look what happens. I'll pull it out.
Bubbly.
Bubble like crazy. Take pictures now. The bubbles are positive, the no bubbles are negative. And now you want to stick that in here. And don't touch the end.
You said bubbles are positive?
Bubbles are positive because it is an enzymatic activity that the organisms produce that break down hydrogen peroxide to produce bubbles. That's the catalase.
Okay, that's what I thought, okay.
Okay? Everybody good?
Yes.
Okay. So now we're going to talk about 3 different plates. And you're going to hold that up so everybody can see it moving over there somewhere so it's out of the way.
Wait.
So here, this is the PEA plate.
PEA plate.
The PEA plate has got phenylethyl alcohol in it. That in itself is inhibitory to gram negatives, but allows for gram positives to grow. So this is a selective plate for gram positives, but it's selected for a short period of time, about a week. After that, the alcohol will volatilize out of the media, and the gram negatives will start to grow. Okay, this is important when you get an unknown, because some of you all are going to need a challenge in your life. You might not know it yet, Harlow. You might not know it, Lisette. You might not know it, Christy. Just picking on people. But you're going to get an organism that's gram negative that is very aggressive. It's going to cover the entire plate. This is the way that you're going to recover your gram pos. Some of you forget, and then you'll be in trouble. Okay? Good? So what you're going to do today is you're going to divide that plate into 3 parts. You're going to put one line Brie, 1 line of E. coli, 1 line of Staph aureus, and 1 line of Streptococcus sanguinis. Okay? And so there, you don't have to do it now. In a minute. When you get over there. Right? But for now, we're just showing you the plate so you know what you're doing. When you come back, it's not positive or negative. It's just growth or no growth. Okay? So if it grows on here, which the do gram positives will, the Staphylococcus and the Streptococcus, they'll grow. But the E. coli is going to be inhibited. Now, if you put a bunch of E. coli on there, like somebody I know, I'm not going to mention any name, you might know who they are, then it's going to look like they're growing, but they're not. So just draw 1 line on the actual media, and then we'll let it sit. We'll come back on Tuesday, and that's kind of close to, that's 5 days from now. So we're hoping, well, we're going to take it out tomorrow and put it in the refrigerator, so it should slow down the degradation or the volatilization of the alcohol. Okay? The next plate, hold it up so everybody can see it. It's beautiful. This is a Myra special. Myra is an expert at making media. So what she did here is she took 5% sheep red blood cell, she ran it through a filter to make sure it's sterile, and then she added it to the media when the media was just about room temperature. The media itself, if you hold it up, it's bright red because that's the sheep red blood cell in the actual media itself. Okay? This is enriched because blood has been added to it, but it's also a differential because we're allowed to see how the organisms affect the blood. And there are 3 possible reactions that occur. The first reaction is what we call alpha hemolysis. And that means that the organism partially breaks down red blood cells, and it results in a green zone around the growth or around the colony. Okay? The second reaction is called beta hemolysis, and that means that the organism breaks down the blood completely, and it results in a clear zone around the colony. And then the last one is called gamma hemolysis. It's just following the same pattern, but there is no real hemolysis. So here, there's no breakdown of the blood, and therefore the media continues to be red. So we say that it has a red zone around the colony. Okay?
And that was the red zone one? What was it?
Gamma hemolysis. Okay?
What was
So
the second one called?
beta hemolysis. It's on the board for you right back there. Right, that thing. So which one indicates that the organism is very dangerous to us?
Alpha?
Oh.
Beta. Because it's complete breakdown of the red blood cells. And if it can break down the sheep blood, it can break ours down too. What you're going to find is Staph aureus is a major pathogen. So, Annalisa, will you volunteer to do a 4-quadrant streak for isolation with Staph aureus on blood auger? Thank you very much. The last plate, no, it's
I
not
know it's
like
not.
blood.
I know
It's
it's not.
kind of an orangey pinkish color. This is mannitol salt agar. Mannitol salt agar is designed for a couple of things. Number one, It has 7.5% sodium chloride in it. That means it has 7.5% salt. That means this is a hypertonic environment and only halophiles are growing. If the organism grows on this plate, it's halophilic. It's one of these organisms. If it doesn't, it's not one of these organisms. Okay? So here you're going to take 3 different organisms. You're going to take Staphylococcus epidermidis. You're going to take Ameluius 4 organisms, Strep pyogenes and Staph aureus, and you're going to put them just one line. Okay? And you're going to come back in 24 hours. If there's growth on here, but no yellow medium, that means the organism belongs to this group. It's a halophile, but it's not fermentive of mannitol. Mannitol is the sugar, the alcohol sugar in here. The only organism that's really pathogenic that will ferment mannitol is Staph aureus. So really this plate is an indicator of Staph aureus. So if we are working in a clinical lab and we're doing lots of different patients, we can draw a little pinwheel, we can put a bunch of patients on one. We come back the next day and we can find out who's got a Staph aureus problem. Okay? Everybody good?
You would put a bunch of patients on one.
I would, because I don't want to waste the plate of one organism. But, Harlow, will you volunteer to do a 4-quadrant streak of Staph aureus on mannitol salt tartar so we can see it in its full glory? Okay? Good? The last thing we're going to do today is called the Staffly Tech. And the Staffly Tech is an antigen antibody reaction. So this is the actual, just the test area. We're going to place 1 drop of the reagent on it. And the reagent has got a little bit of the antibody in it, and it's been bound to blue latex. Okay?
What is the reagent called?
It's called the Staffly Tech reagent. It's the antibody against Staph aureus. Okay? So, Harlow, can I borrow you for it?
The antibody, I guess.
Come here real quick. Okay. So, stand behind me, and I'm going to give you permission to touch my shoulders. So, will you 2 ladies stand up? Okay, so Harlow, put your hands on my shoulder. I am the antibody. I look at the Y. Harlow is the latex. Those 2 are Staph aureus. So when I come in, follow me, don't let go. When I come in contact with them, I'm just going to grab you. I will bind them up like this, and I will have them so that they're bound up to you. And that causes what we call agglutination. Thank you very much for playing. That causes what we call agglutination, and that's positive for a rapid test. Some of you all might have had a rapid test done at like a Minute Clinic or a doctor's office or something like that, but they were mostly looking for Streptococcus pyogenes, the one that causes strep throat. How long did it take for it to happen? Not hours.
It was fast.
How about a few seconds? But they made it, they drew it out a little bit, right? So they went over, they took it, they swabbed your throat, they went to the back, some magic happened. It happened in seconds, but they wait about 10, 15 minutes when they come back because they're gonna charge you $140 and they want you to think that they did something real clinical back there. What they did was this. And so this is the reagent for Staph aureus. So I'm gonna open up another sterile loop, and I want you to grab it from the middle. And I want you to go in and take a little bit of Staphylococcus epidermidis from the actual surface. Okay, that's good. And then mix it up right there really good. Mix it. Like you're making a cake.
Blow a bubble in the
No
hole.
bubble blowing. Okay, good, that's good. Okay, now take it, turn it around, and then I want you to, don't, look,
I just told you the bubble.
okay, now, go in here and grab some Staph aureus at the bottom, right at the bottom. Okay, now, let me see.
I see
Let
it.
me help you. Oh, you have it.
Yeah, I had some.
Now mix that in that other one. There. That's already reacting, I can tell. So then put that in here.
It's full.
I know it. So you can already see the positive reactions, right? Can you confirm that you see little clumps in there? But then you might, okay, not yet. Let me shake it a little bit. The way you do a little shaky poo with this is you just move it back and forth, and you'll get to see the antibody antigen reaction very quickly. So now you can really see the clumping of Staph aureus by the immunoglobulin, by the antibody. And so that Staph aureus is positive, the other one is negative. So how long did it take? Seconds, right? And so it's quite gorgeous. It works effectively. It always works. This is what we do now instead of doing all this other testing. But every single person in clinical science had to go through this stuff to understand that stuff.
Can you look at this on a microscope and look if you wanted to?
Absolutely, you could. Okay, but you can see it right there.
I know, but I want to see deep in it.
You can see what?
I want to see all deep in
It's
it.
gonna be clumpy looking stuff, right? Good. Anybody have any questions?